Okay, so welcome everyone, welcome the ones on Zoom to this third session of the academic training lecture series on open source. We have the pleasure of having here with us Dr. Jenny Molloy from University of Cambridge, and she will talk about open source approaches to intellectual property, so more focused on open hardware, and looking forward to hearing her presentation. Thank you.
Great, thank you very much. So my name's Jenny Molloy, and I'm based in the Department of Chemical Engineering and Biotechnology at the University of Cambridge. So my group is called the Open Bioeconomy Lab, and our kind of area of research is how to build a globally inclusive biomanufacturing value chain. So that's a lot of words, but basically what we mean is distributed manufacturing across a number of countries, and particularly low-middle income countries of biotechnologies. And how we can kind of build those manufacturing ecosystems to be more sustainable, to shorten supply chains, and to enable kind of more agile production to meet the needs of both researchers and innovators and the more general population that can benefit from biotech. So that's a number of my group there who've been working on some of the projects that I'll show, although, to be honest, a lot of this talk is more about the broader sort of ecosystem and movement for open source in biotech. I'll touch on hardware, but also on wetware, which is the sort of biology squishy stuff as well. So first of all, I think I'm the third speaker in this series, so by now I've no doubt that you know what open means, although at least you've had two opinions on what open means. So there's a lot of different approaches to what we mean by open and open source. My kind of approach that I'll be talking about in this presentation is a little broader than the sort of legal aspects of licensing. There is an aspect of universal access, and so this is a framework that was laid out in the sort of ICT for development community. But universal access is really the sort of covers some of the legal aspects of making something open. We'll talk about that more in a second. But a lot of my work also involves thinking about how we move towards more universal participation in producing technologies, so how we involve stakeholders more, and thinking about how to do effective collaborative production. Because a lot of open source projects, there's obviously an aspect of being kind of available to people, but it's also the process of creating an open source project often looks quite different from a closed source project because you have this openness to participation and collaboration. So at a very fundamental level, how do we make technology open? I'm covering this because I suspect that you are more familiar with digital technologies like data and software, which have licenses. And so you may be familiar with the Creative Commons licenses, but this is just one means of basically making a work that's restricted by copyright more open by applying a license that gives some of those rights to other people without them having to ask for them. And so this is very common in the sort of scientific publishing community, in also data, although many of these licenses were not specifically designed for data, and to some extent in kind of documentation of hardware and software as well. But hardware and biology has some additional quirks, which I won't go into in great detail, but just to say that there are, and those of you who are familiar with the open hardware license that CERN has created will be aware, specific licenses for making hardware open source, because the legalities are a little bit more complicated. You've got aspects of copyright, you've potentially got aspects of patents, although not patenting your thing is quite easy, you just don't patent it. But you may have some other patent kind of related considerations, or you could have patented it, but still want to make it available under an open source license. And there's also design rights and other kind of quirks of hardware. And so that requires a sort of separate legal toolkit in order to make things actually open and hit that kind of universal accessibility in terms of the rights to use the technology, or at least the right not to exclude people from using the technology. In biology, we actually have an extra layer thrown in, which is that we have patents, obviously, but if you're talking about a biological material like DNA or some cells, There's also something called material transfer agreements, which are actually contracts. So if I send a biological material, like a bacterial cell line, to a colleague, from my university will often issue a material transfer agreement that lays out what they're allowed to do with that material. And unfortunately, oftentimes, those material transfer agreements are quite restrictive. So even if something, for example, I have some DNA, and the DNA encodes for an enzyme that is no longer patented. It's expired, which is a fantastic way to put stuff into the public domain. After 20 years, the patent expires. And then in all the areas where it was patented, that is now available for anybody to use for any purpose. Even if the patent is expired, they can still slap a contract on it and say, you cannot use this for commercial purposes and you cannot redistribute it, which is very annoying because it means that you can't pass it on to your colleagues. They have to order it themselves from the supplier. So we've developed an open material transfer agreement, which basically offers the same kind of protections to the institutions because they're worried about sending material to someone and it's not what they claim it is, or it doesn't work, and they might get kind of repercussions from that. We keep all of that legal protection language, but we basically explicitly state that you can use this material for commercial purposes and you can redistribute it. So this is just to illustrate that whenever you're thinking about accelerating innovation through making stuff more open, you have to consider your particular context and all the layers of legal stuff that might inhibit you from doing that. And this is kind of one way in which we've worked around that in the bio space. And we have a paper on that in Nature Biotechnology that you can check out. But basically, we've sort of covered a lot of the same principles that you'll find in open source hardware and software, licenses or other legal documents, so that the access is free of any royalties, that there may be attribution, that materials can be reused, they can be redistributed, and there's no discrimination against who can receive those goods. And so this is all kind of very much in terms of the principles of openness to get around this very specific problem. So once we've got around this idea of making something legally open, why would we bother doing that? So in what context and by what mechanisms can open source sharing actually help generate impact? I think these are general towards other open source technologies as well, but it's a way that we think about things in terms of the biotechnology space too. So you might apply open source approaches because you want to accelerate research and development. And so one of the key proposed benefits of open source is that reduces friction in access to knowledge and materials. And we've certainly found that, for example, in terms of the open material transfer agreements, a classical material transfer agreement for some institutions can take weeks to months to process. And so by making this that kind of standardized form that is much simpler, that really can reduce the friction there. Also in terms of simple things like data and software licensing, people arrive at a website, they immediately can understand what they can and can't do with that knowledge and those tools, and they can go ahead and do it if they're able to. They don't have to kind of necessarily contact you. It just reduces all of that. So there's an inherent acceleration in reducing those transaction costs. But the other aspect is, if you want to have user innovation and kind of people contributing ideas back to the pool or expanding your pool of developers, then open source, for kind of similar reasons in some cases, gives you more ability to do that by reducing that friction. But also, if you're taking the approach that openness goes beyond the licensing and that it's actually about building a community or a collaborative development structure, then that's a really good way to get that user innovation. It also kind of frees you up in a way to focus on what value you're adding beyond a kind of restricting something by intellectual property. So thinking about what are the aspects beyond just the technology. So at least in biology, there's an awful lot of know-how. So it's not just that you can present somebody with a software script or a data set and they will kind of intuitively know how to do that thing. You can write down a biological protocol in incredible detail and it might still be challenging for someone to actually implement it. So we have a lot of kind of these sticky, intangible knowledges that need to be passed on and skills. And so that is kind of something that we would dedicate more time to, having made all the other stuff openly available, thinking about collaboration and community, using the technologies, building trust and buy-in. But there's definitely reasons to not use open source approaches. And this has different implications for different, or why you might consider not using open source approaches. So in many technologies, there's a significant need for capital, and I think biotechnology is a very good example of where this is true. Many biotechnologies might take 10 years or more to reach a point where they're available to be marketed. I worked for my PhD for a company that genetically engineered sterile mosquitoes, and it was 11 years from the founding of the company to them actually having a commercial release license. So that's some patient capital that was required. And so if you have no ability to create a monopoly, that could impact your profit margins or your ability to maximize profits, and it could impact your ability to get investment. There are many ways kind of around this, but I think this is particularly true in these kind of longer term technology fields, that it's probably not the way to maximize profits, which doesn't mean that it can't be a route to profit, but monopoly is very effective at maximizing profits. That's kind of what it was designed to do. So if that is your goal, then you probably don't necessarily want to go down an open source route. But I think many people who are exploring these options, the absolute maximization of profit as the only goal is not the entire story. So if you need to, for whatever reason, defend or control your IP, then you might want to restrict it in some ways. An example of this is there's a company that makes basically a nutritional substitute that's used, that they want to manufacture in low middle income countries, and they actually use their patent strategically to prevent lower quality manufacturers using their manufacturing protocols, which is only as effective as it's enforceable. So it's a little bit unclear how useful that's been to them, but there are definitely ways in which people kind of use patenting strategically that's not necessarily to do with restricting access to the knowledge, but more restricting access to the ability to make something or trying to control quality. There's also an argument for not necessarily using open source approaches where it absolutely won't be used, because so this is very common in the open science field. So the Royal Society, I think now over a decade ago, wrote a very influential report on open science where they talked about kind of particularly open data, where they talked about intelligent open data. So really thinking through, if this data set is genuinely only ever going to be used by me, and it's going to take me, 3 months to make it available to other people, that's probably not a smart use of my time and, public funding or grant funding, so let's not do it. So it is kind of thinking through, what's the balance, the trade-off between, the effort needed to document something effectively and its likelihood of being used. So that's definitely something to consider, and is also a tension, of course, as we have more and more open data mandates and policies coming through as to how you deal with that trade-off. I, of course, you might have guessed, am a massive advocate for making stuff open wherever possible, but I still think if you're convinced it's going to be a waste of time and you're the expert in that, then it's probably going to be a waste of time. So these are some reasons why you might not use open source, but it's not a massive list. And so I think the argument I would make is always just really considering the context, and that's something that we have to do a lot, particularly in the sort of biotech space, where we have these sorts of very complex technologies that are actually like multiple processes and multiple biological materials and multiple protocols sometimes. So in terms of so we've made something available, we've considered why we might make it available and what our intentions are. So once we've done that, what are effective collaborative engineering approaches that we might take? So in a lot of our work around the biotech space, we try to think a lot about collaboration and co-creation. So this can be challenging because you need to, if you really want to do those sort of second two layers of universal participation and universal kind of collaboration, you're going to have a coordination challenge of people and things. And this is much more true for biology and also for hardware than it is for software and data, because software and data is digital. You can ping it across the world in milliseconds, whereas for us, we have to actually ship stuff, which can be a real challenge. Like right now, I really at some point today need to set up a shipment on DHL of a whole bunch of consumables to to Ghana for a field trip that's running in a couple of weeks. And next week, I have to ship a whole bunch of stuff, probably about 50 kilos of resources to Kenya for a training course that we're running. And that actually takes quite a lot of time to organise. So there is a sort of coordination challenge. And if you're wanting to co-create, you often want to be working on the same thing together. So being in the same place is very useful for collaboration around hardware and biology. But as we know, it's not always possible, and trying to kind of get this stuff to multiple parts of the world so you can work on something effectively together is more challenging, I think, in this context than digital technologies. We work a lot with low middle income countries, so there's also, for us, perhaps more than other projects, a challenge around navigating different levels of resources, different ability to kind of shape the project, and different collaborative approaches. Some academic environments are very hierarchical and others aren't, so even that can be a challenge sometimes in terms of, certainly in the UK, it's a fairly flat system, I would say. There's an informality around students talking to lecturers and researchers, and there's a sort of very team environment, at least from my experience. That's not necessarily true in other countries where there's a much more of a hierarchy. So that can be a challenge as well when you're trying to collaborate on an open source project or any project. But I think the point I'm trying to make is that in open source, if this is a real intention of what we're doing, this kind of becomes more of an issue. And then also documentation. So months can get lost to poor documentation, and particularly documenting know-how is very difficult. So we try videos, we try lots and lots of pictures of what to do, but it still sometimes is not enough. And we've had issues that have been, plaguing projects for 18 months, and they've only been solved basically when we were able to go in person and just watch someone doing a thing. And you quite often find this as well. Sometimes I just have to go into the lab and watch my student doing a thing, and then I'll figure out what's going wrong, but we could have spent an hour chatting about it and not really worked it out. So I think one of the key things that we try and do, I talked about accelerating transaction costs and kind of effectively enabling people to take your technology and work with it and contribute to the project. A lot of that, I think, is really reducing kind of dependencies. So enabling use of your technology, ideally without you, as much as possible. And so in my context, that basically means that when we're working with scientists in low middle income countries on biotechnologies, particularly diagnostic development is a thing that we do quite a bit, and I'll talk about it a bit more later, but that we don't just want to create a dependency where we're using some fancy biotechnology technique and I have to keep repeatedly shipping it from Cambridge, because that is not really what I want to do day to day, but also because it doesn't really build capacity and capability or independence and autonomy. And so we think a lot about not just kind of, creating a technology, but also the process. So how do we produce and have a cycle? So to give you a hardware example, in the 3D printing world, there's a project called RepRap, which some of you may have heard about. It's an open source 3D printer. And the thing that captured people's imaginations with RepRap was the idea that it's a printer that prints itself. So you obviously not all of itself, there are metal parts as well, but you basically can print the components of the RepRap on a RepRap, make more RepRaps with some additional bits and pieces. And so you kind of have this, you have a means of production for the means of production, and so it goes. And so we try and incorporate that when we're training. So we have a whole open source collection of DNA to make enzymes. I'll talk about it a little bit later. And when we run training courses, we're basically always teaching people how to make the enzymes that they need to work with that toolkit, so that they're able to basically incorporate their own biological components into our toolkit and keep going. So by the time they leave, they're able to basically just drive things forward themselves. They still need to import various things, but the things that they need to import are significantly less expensive and less difficult to ship than the the end product. So it's trying to think about the sustainability of that and give people the means to kind of keep a project going. Also considering kind of what the end game of a project is. Often in open source, and I'd say particularly in academic projects, this is a challenge that we face. People make very, very useful projects, but then they sort of die. We're currently undergoing we're building a compendium of open source projects that have come out of the University of Cambridge at the moment, because we want to kind of explore some of the potential impacts and where they've been adopted. And I think a big takeaway from that is that a lot of the people that we're contacting, even with projects in the last 2 to 3 years, the projects are no longer active because someone moved on or the funding was not sustainable. And I think this is a systemic issue, but if you can build it in at the start of your project to reduce the dependency on a single grant or a single person, then that's really helpful. Because we all kind of have the dream that if we put our project out there, someone will take it and do something wonderful with it. And that might happen, but maybe not for some time. So I think you've heard quite a bit on this set of meetings, or will hear about kind of, storing things for the future and how to kind of create, reworkable work. And so that's kind of where we're going with this. And for us, again, just a kind of similar point to what I made earlier, it's really kind of balancing the local autonomy of the people that we work with to kind of build out biotechnologies independently, but also acknowledging the reality. We live in a global world, and that they need to kind of definitely get some stuff from other countries, and also knowledge. It's a bit tricky. Like, on the one hand, we want people to have the agency to basically independently create research agendas and move them forward. But on the other hand, international collaboration is always enriching and rewarding, but it's more about, for a lot of the researchers that I work with, international collaboration is more of a necessity that they can't do without, because without international collaboration, they wouldn't necessarily have the links and the resources to do the research in the first place. So I kind of think about what we try and do in our projects is work towards that being a positive and intentional choice, that they want to work with me rather than they have to work with me, because otherwise the work would not succeed. So that's sort of fairly high level. So I want to kind of give you a specific example. So we've worked a lot on access to reagents through local production in Cameroon, and I'll just talk you through why that project came about and the problem it's trying to address, and then some of what we've done so far in terms of building out the open source technologies, but then also thinking kind of what the future might hold. So there is a real issue with the supply chain for laboratory reagents. I'll show you an example later of PCR, which, 2018 when I started this project, not that many people outside the biology space had necessarily heard the word PCR repeatedly, but after COVID, I think we all have. So PCR is a technique that we use for diagnostic tests, and it needs a lot of reagents. We'll talk a little bit later about exactly what they are, but the point is that most of these are not manufactured, or manufactured in only small parts of the world. So this map, the darker blue is the higher levels of export of laboratory reagents, and the lighter green, the least export of laboratory reagents. Export is not like a perfect proxy for manufacturing, but it's fairly good. So as you can see, most laboratory reagents are exported/manufactured in North America, Europe, and parts of Asia. But there's very little in Africa and quite minimal also in Latin America. So this is a challenge for a number of reasons. And the kind of effects of that challenge are seen in the sense that there is less research output from for a number of reasons that are not just related to the supply chain, but it is a real impediment. So if we look at, say, synthetic biology, which is the field that I work in, 24,000 papers can be found in lens.org, but only 1,128 at the time that I got this statistic are from low middle-income countries. I exclude China, which is technically still a middle-income country, but is actually the third most prolific synthetic biology publisher, so I've removed them from the equation. But for everybody else within the low middle-income bracket, it's not a lot. So there in terms of biotechnology, there's definitely a disparity in terms of who is able to use and publish kind of biological engineering work, which is not to say that other forms of biotechnology are not well represented in those countries, but the engineering biology aspect is definitely less represented. This can also have an effect on innovations. This is a map of COVID diagnostic innovations. This is in 2021, so this is basically the first innovators, right? So this is who got tests produced, or rather developed and produced, and on the market quicker. And you can kind of see the pattern repeating itself, right? So there were some tests being developed in parts of Africa, particularly South Africa and Egypt, and in some parts of Latin America. I think this map got populated as it kind of post this map being produced. So there are definitely more tests that were made in Latin America and also Africa, but there's a very clear disparity again. You can see which countries were faster at doing this, and this is partly related to infrastructure, but also partly due to supply chains. I was in touch with a researcher in Costa Rica who really wanted to develop not so much the diagnostic tests, but the RNA extraction techniques, and they waited over 2 months for delivery from the United States of some of the chemicals that they needed to do that work. So it plays into this pattern. I'm not claiming that supply chains are the whole cause of this pattern. There's a lot of other issues. But it also impacts on health. So this John Knegsong, the director of Africa CDC, talks about how Africa had funds to pay for reagents, but at the height of the pandemic, it couldn't buy them because basically the countries that were manufacturing them were not exporting because they had the need themselves. And so you have these kind of issues with dependency on supply from other places. That particularly, this was a very extreme case, right? The COVID pandemic was a very extreme situation, but the impact can be seen across research, across innovation and the speed of innovation, and also across direct access to the end products and the health products. So I think that this idea of being able to be more autonomous and have more manufacturing capabilities in regions has become more of a political thing now, particularly around vaccine manufacturing, but also in diagnostics for the reasons that we just talked about. But there's good news as well. So a lot of the challenges that were talked about in COVID around intellectual property, particularly for diagnostics, less so for vaccines, but a little bit, are very different in the Global South compared to, say, the United States. So in patent law, you have to pay to register a patent in each and every single country/region that you want the patent to hold. So if I pay in the United States and in Switzerland, I can only protect my invention in the United States and Switzerland. Somebody could make it, sell it, do whatever they liked in South Africa, and I would not be able to pursue them for that. That's how patents work. And so 99% of patents have rights in fewer than 20 countries, which means there's a huge freedom to operate, particularly in the Global South. Only about 1% of patents are in fact registered in the Global South. So as long as countries are manufacturing and selling locally and not attempting to export to the US, where they are protected, then there's a huge opportunity here to kind of move forward with technologies, even when they're not open in other places of the world. So this is not quite open source per se, in that it's not intentionally made open to everybody, but I think it's just useful to kind of put this point in the chart so that you kind of see the broader landscape. So back to PCR, just so I can explain very specifically the challenge that we were trying to overcome in Cameroon, or at least a pilot to try and overcome it. So PCR is a fundamental technique in molecular biology. It takes a small amount of DNA and it multiplies a very specific piece of it, so you've got lots and lots of copies. And once you have lots of copies, you can see it. For example, here, you can see what have you got the DNA, what size is it. So it's used for testing. Not necessarily that you have to run it on that. There's instruments that can monitor it in real time, and that's what they were using for COVID tests. But once you have a lot of it, you can also engineer it, change it. So this is fundamental to making vaccines. I talked already about mosquitoes that I used to work on, where we engineered them to be sterile, which meant that they kind of brought, lower the population of mosquitoes and block the transmission of dengue, or reduce the transmission of dengue. You also need this if you want to edit or engineer any crops. So it's basically used across pretty much all biotechnology labs. And what it looks like is this. This is someone putting together a PCR reaction. This is like 90% of molecular biology. It's basically moving small amounts of colourless liquid into tiny tubes. That's pretty much how my PhD students spend their day. And so what's in the tube? We don't know because it all looks the same. But it's basically the DNA that you're interested in, some buffers to make the pH happy, some smaller pieces of DNA that basically target the bit that you want to increase in number and amplify. And then you've got DNA polymerase, and that's an enzyme, and that's the kind of protein that actually copies the DNA, and it builds it up through DNTPs, which are basically the ATCs and Gs that make up so you've got your building blocks, your ATCs and Gs that are called DNTPs. You've got DNA polymerase that basically runs along the DNA and matches the ATCs and Gs to make more DNA. And you've got the DNA. So that's pretty much what we need. And what's the challenge here? Well, the supply chain for these reagents is usually involving a cold chain. And to go back to our comment earlier, it's got to come from here, here, or here to say here on ice and arrive on ice, not defrosted. This can happen, but the challenge is it takes ages. So if I order some PCR reagents that arrive looking like this, in Cambridge, I typically get them in about 2 days, and they are definitely frozen. We have no issues with cold chain. If my colleagues in Cameroon order them, probably around 4 weeks, and they may or may not get out of customs frozen, because customs do not necessarily understand these things. They may not necessarily even have a freezer. There are some researchers in Zimbabwe I was talking to where their local airport definitely doesn't have a freezer. So as soon as they know that something is coming, they have to go directly to the airport to make sure they immediately get it off the plane effectively. Otherwise, it will be ruined. So these are some of the challenges that people have. And so it's not necessarily just an issue of can we afford it, it's also an issue of the time. Like if you try something and it doesn't work and then you have to wait another 4 weeks to order some more materials, that's a very, very slow innovation and research cycle compared to being instantly able to update some code and debug some code or rapidly try things out. And if you're really innovating, then your chances are you're going to fail quite a lot, so this is going to be a common occurrence. And so, this is Lenshina Agbour, who we worked with in Cameroon to set up a lab in 2019. She's in the lab there, and she's now a PhD candidate in the UK, but has talked about, the gap between what she experienced in Cameroon in terms of being able to do her research and what she's now experiencing in the UK couldn't be wider in terms of the access to the tools to do that research. And she was working on malaria infections in pregnant women. This is Stefan Verdanker at the front here, who did a master's project looking at sustainable solutions for nematicides. These are parasitic worms that are basically pests in banana plantations, and they're currently addressed chemically. But he was looking for a sort of bio-alternative and waiting for reagents to be delivered from outside the country, and delays with all of that basically added a year onto his master's project. So these are kind of very, very, kind of extreme in our eyes, but quite normalized experiences for a lot of the people that we work with. So I hear you ask, how can open source play a role in this? So our goal is basically increasing resilience and autonomy in terms of access to research tools through a more distributed supply chain. So I've shown you the map, that now it's pretty centralized. It's manufactured in a small number of places. It needs to get to a large number of places. So if we can kind of redistribute that network somewhat and have local manufacturing close to the point of use, the idea is that it could reduce costs. It may not, it really depends on the context. But it will certainly reduce the time to delivery. It will remove the challenges with getting stuff through on a cold chain. And so in order to do this, we've developed some technology, we've built communities, and we've thought about kind of enterprise. So how do we really deliver access at scale? I won't talk about all of these activities because we don't have time, and I want to leave time for questions, but I'll run through at least the technology stuff. So what we made was something called the Open Enzyme Collection, which is it's DNA. So it's pieces of DNA that encode a bunch of enzymes that are very useful for molecular biology, and also a lot of other pieces that you need to make those enzymes in bacteria and purify them. So you don't need to know the details, but basically we have a number of these different types of DNA that you can put together like Lego blocks to make a longer piece that will make your protein that you want in a bacteria. So we make a circular piece of DNA that has all these pieces in it of your choice. You pop it into a bacteria, and the bacteria will start producing your protein, and then you can kind of purify that protein and start using it for your experiments. And we did a sort of second round where we took the ones that were very related to diagnostics and made this very specific collection, which we've now put under an open material transfer agreement. These numbers are actually a bit out of date. I copied in the wrong slide. So at the time, it was in 185 labs in 41 countries. Between this collection and the kind of broader collection, we're now in over 500 labs in about 50 countries. And this gives people, if they have this and some bacteria, they can basically make 20 different enzymes, give or take. Some of them are more difficult than others, so you do need more materials. But the idea is that this is available, and it's also customizable, right? So part of the value of having something that is openly shared and someone has thought about the idea that someone might reuse it is that it's been designed for reconfiguring. And so this here, all these kind of overlaps, that's basically they define the connections between the Lego blocks. So they make it much easier to recombine stuff very quickly without having to order a whole bunch of connecting pieces. So when we distribute it, it already comes with all of the little kind of connectors that it needs, so you can effectively biologically snap stuff together faster in a Lego style. Although Lego is not a great example because famously they're quite universal in their connectors. These have specific connectors so that they always turn up in the right order. But we can talk about the biology more at the end if you like. So we've got all this DNA. It is open source. It's distributed widely under the open material transfer agreement that we discussed before. But we also need to think about the process for using it, because in many cases, people do not have access to the lab facilities that we do in Cambridge. And so I'm just going to give one example of a sort of small project that we did. So we normally add, this is how we grow our bacteria, sometimes, in these flasks. So this is a rich medium with sugars and proteins and other things that the bacteria need. And normally, when we want them to start expressing the protein, we add a chemical called IPTG. And we were having trouble with IPTG because it's kind of stable, but also not very stable. So we were having to ship it repeatedly to Cameroon, again, dependency. And so we basically thought, well, IPTG is basically an analog of a type of lactose. So we thought we'll go old school, the way they did in the 80s, and use lactose instead. And this turned out to work. And so we thought, well, should we just use milk? And so we tried using whey that we made from milk from the supermarkets, and also boiled milk, which was effective. So you can see this is our protein. It's here. It's not here before we add the milk. It's here after we add the milk. But there's an issue here. So when you add milk, your broth goes cloudy, but that's also how you tell there's bacteria in the broth, is that it goes cloudy. So it kind of obscures something that we need to know about. So we thought, okay, we're not going to be able to tell if our culture is contaminated if we put milk in it. So instead, we can put it on a plate like this. So now how we routinely make our proteins is that we spread the bacteria on these Petri dishes, and this agar here contains the milk, and it goes a bit cloudy, but that's okay because we're just spreading the bacteria on it. We don't need to be able to kind of look through. We use the passage of light through that liquid to figure out how many bacteria we've got. So that's why the milk is not only checking for contamination, it's also that some of our measurements will be really off if there's milk in there. So here we just scrape off the bacteria after leaving them overnight. And then we put those in a tube and we can purify the protein or do whatever we need to do. It's super easy. We don't need a big centrifuge to spin everything down. Goes straight in the tube like so. Ta-da. And I think I have to click one more. And from one plate we get thousands of PCR reactions worth of enzyme. So thousands of PCR reactions worth of enzyme would be on the order of like minimum 100 to 200 pounds, probably Swiss francs. but it depends how much we grow. I mean, you know, you can keep scaling it. So this is basically an extremely cheap way of getting quite a lot of enzyme. And so you can see here, there's no enzyme, lots of enzyme. So we have these protocols and we share them locally via protocols.io, which is a site online, so anyone can access them and try them out. And so the vision for all this technology piece coming together is that someone somewhere could find a really cool piece of DNA in some local hot spring or some kind of interesting environment, pop it, make a Lego block, put it into our collection of Lego blocks that will add all the other functions that it needs. And then we are also trying to develop protocols so that they can rapidly kind of assemble and test those things. Again, all using either open source, low cost, or open protocols. And this is quite exciting right now because biology is like getting easier to do. I mean, the lab work's still hard. Biology has a mind of its own sometimes, but we've now got much better machine learning models for all this sort of stuff. We've got amazing abilities to kind of look at sequences from across the world and try and design new and exciting enzymes as well. So we're really interested to kind of integrate this sort of the open source digital technologies that the bioinformatics community are making and the open source hardware, and have this cycle where we can really have a kind of interesting discovery, innovation and manufacturing cycle that all happens in the country of origin of the biology, which is not how it works right now. Usually someone finds something and then they would ship it to a collaborator in the States or the UK to figure out, right? Which is not ideal in terms of equity and equitable capacity building. And so they were trying to subvert this in a very small way, and hopefully we'll get there. We'll see. So I have some quick fire case studies for the next 5 minutes, and then we'll have 10 minutes for questions, if that sounds okay. So that's an example from my work. In terms of biotechnology, it's a tough gig for biotechnology and open source for the reasons that I mentioned before, which is that you need a lot of capital. And one thing investors do not like is non-protected IP, typically, and particularly in the biotech sector, it's very expected that you will have protected IP. So it does make some of the arguments for getting that capital more challenging, I think, than in other sectors where the kind of patents are not as seen as valuable, or there's other reasons why there's less of a focus on that. There just really is a big focus. However, there are some companies that have managed to scale with an open source business model that work in biotech. So an example is Opentrons. So this is an instrument that I showed you on the last slide, actually, that we use in our lab. It's a pipetting robot. As I mentioned, 90% of our work is moving tiny bits of liquid around. We are effectively human liquid handling devices most of the time. So this is a way to automate that process. And it's basically, instead of a human holding these pipettes, it's a robot holding the pipettes. So this was actually originated in a community DIY bio space in the US. So it was basically a real garage biotech startup. This is the first version 1, which I do have one of in Cambridge. And this really was basically like a 3D printer type of chassis with some pipettes. And these are actually just pipettes like from the lab. You can see they've just slotted them into the holders there. And so they started off with this. The latest version looks more like this. So they've really professionalized. They've gone to a model that is now kind of, as you can see, manufactured professionally as opposed to sort of put together. They still keep the sort of base designs and for the hardware, as in the electronics and the software available. But for obvious reasons, this is not something that you can build in your garage. This is kind of manufactured more at scale. So some people in the open source community would argue that they have moved away from the open source ethos, but they still, I think, would very much count themselves among the open source community. They do make a lot of their stuff open source, and they do have a real focus on community building as well. So we have one of these in my lab, and I think we have most of these add-ons at the bottom here. They, as you can see, have all of their software on GitLab, lab robots for biologists. And they did a lot during the pandemic in terms of setting up huge banks of their robots to automate sample preparation. So this is basically an OpenTrons robot preparing RNA extractions from COVID samples. And so because they're reasonably inexpensive, they're less than $10,000 per robot, which is cheap for biology instruments. They're able to make a lot of them. I mean, you can see basically there's an entire room full of them here pretty much just doing this. And it vastly accelerates the time that it would take for a human to go through all of this stuff. So you can see the human is doing the barcode scanning, and then they'll put it on the robot. You can see here there's basically just rows and rows and rows of the robots. So this idea of kind of using these pretty low-cost automation methods, but scaling up in this way, really caught on and has been very successful for them. So they closed a $200 million Series C, and to build out their robotics, diagnostics, and cell engineering services, as well as providing the robots for researchers. So it's a good example of a business model, I think, that was definitely originally based on open source, may have moved away from some of the purest principles of open source, but I think it's still, this diversification to offering services has a bit of a parallel to the open source software industry as well. Another two quick-fire examples. So Tesla, some time ago, made a patent pledge to not enforce their patents, which is slightly different from being totally open source. But they basically made this promise that they would not go after other electric car manufacturers who, and this has sort of changed a little bit since, but the basic principle still stands that they wouldn't pursue and enforce their patents. This might sound like a very odd move for an automobile company to make, particularly one like Tesla, which is very tech-driven. I mean, sometimes, if I'm lecturing to students, we do audience participation at this point, but I won't put you through that. But have a think maybe about why a company like Tesla might do this. They've never really publicly made a very categorical statement as to why they did this. But the general feeling among people who study patent pledges and why people do them and motivations is that the electric car industry has a major dependence in that the electric car manufacturers are not going to be putting in all of the electric car charging points. They're dependent on public funding and other kind of governmental incentives, basically, for electric cars to take off. Now, in order to kind of get to that point, there needs to be a lot of electric cars of varying levels of affordability. There needs to be a big industry that can be a strong voice to have that infrastructure put in place. So by making their technology more available to other electric car manufacturers, there is a potential that they can accelerate the industry and actually build, so they kind of expand it and perhaps get a slightly smaller kind of slice of the pie, if you like, but of a much bigger industry, because they've been able to kind of solve this dependency on effectively collective and in some cases publicly owned infrastructure that Tesla as one company alone probably couldn't argue for. So I don't know, that there are probably other reasons, and that's just one of the sort of hypotheses, but to go back to our theme of dependency and how open, you might think about making stuff open and accelerating collaboration and technology development, I think this is an intriguing example. And finally, a pharmaceutical example. So the Structural Genomics Consortium has been around since 2004. It's a huge multinational effort to basically figure out the structure of every protein in the human genome for the purposes of drug screening. And they figure out the structure and they make them all available totally openly. And they also do a number of kind of drug screenings that they make the data available for. And a lot of their funding comes from the pharmaceutical industry and not the usual suspects for open science. So they get a lot of money. Like each person, I think each company, and forgive me if I've forgotten this number, but it's on the order of millions of dollars. So I think buying into the SGC is around $7 million a year, something along those lines. But I could have misremembered that one. But they basically get a huge amount of funding from the pharmaceutical industry, and you might think why? Because they're just going to make this stuff and put it available. Why would multiple companies that are usually rivals, Roche, Glaxo, etc., put money into the same open science project? And the answer is because a lot of the failure of drug discovery happens much earlier in the process. Like 99% of stuff fails very quickly. And as you move through kind of different tests and particularly clinical trials, you start dropping off the kind of higher risk compounds and candidate drugs. And so for the companies, it's basically a de-risking approach, so the SGC does the bit that has a high failure rate, and because they're all pooling into that, the alternative would be that each of the companies independently does this very high risk research and redundantly and independently has to throw out 90% of what they've done, as opposed to the SGC kind of getting rid of the bad candidates first and then ending up with a pool that eventually ends up in the companies, right? They do the last piece as a kind of proprietary thing. But what the SGC has done is basically the pre-competitive space for the drug industry. They mostly work on very tough problems, I should say, like neurodegeneration, that the pharmaceutical industry has had challenges in making any headway. So they start, the status quo might be that you do very, very, very basic fundamental research here, pre-competitive, and then there's a wall, and then it all happens privately in companies. And the SGC have just kind of been moving that wall so that more and more stuff happens in an open science. And then by the time you end up with some candidates, they're massively de-risked and they're much more likely to actually be successful in clinical trials than any given candidate at the start of the process. So they work really globally. They have a zero patent policy, which they have kind of claimed makes it very swift to establish these collaborations. And they've also spun out their own company. So Medicines for Kids is a company that is developing drugs, particularly for childhood cancers, and they do not patent anything. And so their model is basically they get a lot of funding from patient groups. This is a very tough challenge and not one that Big Pharma has solved. And so they get a lot of funding from patient groups and philanthropic sources, but their goal is to be a sort of proper company, but they just don't use patents. They do use something called market exclusivity, which in the US is where once you get FDA regulation on your drug, you have exclusive rights to market your drug, but that lasts for less time than patents, and it's specific to the US. So they wouldn't patent it, which would mean that it could be produced in other areas. This is still an experiment, I would say, right now, so we'll see what happens, but they're making good progress in terms of their candidates and kind of moving stuff through the pipeline. I'm going to skip the rest of this because it was just a little bit about some policy work. But yeah, I would love to hear from you, any questions that you have, and thanks for your attention. It was a bit of a whistle-stop tour, but hopefully interesting. Thank you.
Thank you. It was super interesting for me, so it's I think also for others. So are there questions in the room or on Zoom? On Zoom you can raise hands and I can just let you speak. So we already have well, okay. Okay, let's take this from SA on Zoom.
Hi.
Hi.
Sorry. I was wondering, like you mentioned, that in the global south, these patents are not always filed and there is sort of unwritten freedom to explore. So I'm wondering, like in software and in electronics, for example, reverse engineering is a very effective way to make advances in this kind of circumstances. So is that also an effective strategy for biotechnology?
Yeah, it can be. I mean, certainly you can often work around quite a lot of biological patents. The type of work that we do is often using we pretty much focus on expired biological patents to just ease the legal side of things. But there is a vast array of different options out there. So yeah, there's definitely workarounds. I'd say a little bit less maybe reverse engineering in the sense that we typically don't have the same kind of ability to reverse engineer a biological system as you might do with kind of deconstructing a hardware device. But there's definitely a lot of working around that you can do and just kind of finding holes in the patents if there's a patent in place. But as I say, I mean, pharmaceutical, like small molecule pharmaceuticals, biologic and vaccines are very globally patented. As soon as you move outside that space, it gets a lot more open. So in diagnostics, for example, we're still finalizing the data on this, but we did a search. So probably many of you have heard of CRISPR, which is this very exciting genome editing technology, but can also be used for other things. So the use of CRISPR for like gene editing is incredibly widely patented. Probably in terms of like biotech patenting, it's one of the most broadly patented things outside of pharmaceuticals. But the use for diagnostics, we really couldn't find that many places where it was a thing, which is good because we're making a CRISPR diagnostic with some collaborators in Cameroon, so that's ideal. But yeah, I think reverse engineering less so, but definitely workarounds. And for hardware devices, which are also very important in biotech, the reverse engineering approach can also work. And then you kind of have to have a hope that you can kind of recreate the functions that they managed, but it should be possible.
Thank you.
Thank you.
Yes, I have two questions on the analogies with open source software. As you know, in many of the open source software is like a reference that we are aiming at to reproduce the success of open source. So the first question is about what would need to happen in open biology to get this kind of, I would not say maybe growth is too strong a word, but to mimic the success of open source software. I remember in a few years ago there was a talk in the Open Hardware Summit that dealt with what would need to happen in the hardware realm to mimic the success of open source software. There were things like composability. Today, if you want to take the software of somebody and add value on top of it, it's extremely simple. And if you have hardware schematics, it's not so simple, right? So is there something in the life sciences realm that you think would need to happen to unleash even more sharing and more creativity? Is there like,
Yeah, I think there is definitely. So this is, if I just flick back to the slide on the toolkit, because no, it was before this one. Before that one, this one. Yeah. So so the field of synthetic biology is kind of trying to do that. It actually grew out of electrical engineering and computer science. So some of the very early labs that describe themselves as doing synthetic biology were out of MIT, electrical engineering. They set up a biolab in electrical engineering, which is quite interesting. But so their goal, and that field's kind of goal in the last, say, 20 plus years that people have been describing themselves in that way, was to apply formal engineering principles to biology and do some of what you're saying, like standardization, thinking about modularity, thinking about kind of good documentation of these Lego brick parts that you, but the kind of big aspiration, to use a word we were thinking, i.e. unrealistic, was that you can basically program biology like you can program a computer, and that you can treat DNA as source code. And so that has been happening. It's not like it hasn't been happening. And I'd say one of the things that we're seeing now is that the technology is catching up with the aspiration a bit. So in the beginning stages of that idea of how can you really design biology, is really difficult. Biology is super complex, and just there's a whole bunch of feedback loops and things that we don't understand. So, I mean, basically, you can kind of make your lovely DNA Lego brick block and stick it in one bacteria, and it will work perfectly. And it might work perfectly in the second bacteria and the third bacteria. The chance of it working across multiple, more than that, is very slim. But it was this aspiration of, can we actually, so the initial switch that you need to compare it to software is, can we actually design and engineer this stuff? As opposed to the point about reverse engineering, basically what we used to do in biology is break something and then try and figure out what it did. So at a DNA level, and I'm very DNA biology based, so forgive me, there are other parts of biotechnology that we're not touching on. For example, we've got fruit flies and we want to understand how does the fruit fly know where to put legs and wings in the right order? You irradiate some fruit flies to break their DNA, and you pick the fly that's got, a leg on its head, and are like, okay, let's see what's going on here. And then you figure out how you can pattern. That's how we used to do it, right? Very brute force, kind of break things, figure out, reverse engineer it. Now the idea is that you can forward engineer. And the challenge that we've had with that is having enough comparative material, but now sequencing DNA is really cheap compared to what it used to be. So we have millions and millions of DNA sequences, probably more than people can really handle right now. The other challenge was that it was very time consuming to change the DNA do anything to change the DNA. So debugging, for example, that you might see in software, you can't really do that well in biology. But now it's very cheap to write DNA as well. Like we can now I can sort of type some C's, A, T's, and G's into my computer, and within 2 weeks, I'll have that DNA on my bench for $100, $150. Even 5 years ago, that would have been like 10 times that potentially. So we're kind of catching it, and then also the machine learning models and kind of AI that are coming in that are helping us do that stuff as well. So I think biology is becoming more malleable, but we do need better technology to deal with the complexity, and we ideally need that to be accessible, but also kind of usable by the average biologist. So I think what we saw also in software is that it became much more democratized, right? So now you much more would find kind of kids learning coding very early in school, a lot of support for how you kind of develop those skills, like in hardware as well with Arduino and the Scratch languages, and how to kind of give that onboarding. We don't really have that in biology. So I think also it would help to get more people involved if we had these sorts of stepping stones. And I think, again, we have something called the iGEM competition, which is a big student competition called the International Genetically Engineered Machines. And then 375 teams every year get together from high schools and universities and design bacteria that do cool stuff. Like, I don't know, they design, biosensors for lead, or they design bacteria that kind of you can shine light and they produce pigments, so you can make photographs on your bacteria, all this sort of stuff. And that's been going for a while, but I think we're certainly we're not seeing it at the level where you can do stuff in a reasonably limited lab setting in anywhere, which is kind of would be something that I think could help sufficiently. Like I'm not saying anywhere, but like at least in research institutions across the world, that we have the point where we can do some real engineering with this sort of technology. And I think, like, because with software you can just grab a computer, but I mean ultimately you have access to that. It's a big difference. So I think to move forward, we really do need the enabling technologies that allow that democratization to happen. That was a very long answer, but does it make sense? Yes. I just think they're like fundamentally different in terms of their ecosystems and requirements. So it's a bit challenging to compare the two. But hey.
Yeah, thanks. I think you partially answered it now, but the question was like, it was quite interesting to see like the way you try to distribute the reagent production everywhere. But then the question is how, what about the distribution of the creation of the original DNA? Because I understand like, you create it centrally, but then you distribute them, but then can they replicate it locally, or do they have to like acquire it centrally? And do you see the production of that original DNA be able to to be distributed in the near future or something?
Good question. So if it's DNA that already, so we have one advantage of DNA is you can ship it not on cold chain, you can dry it down, it's very stable. This is how we managed to sequence Neanderthal genomes, right? It just stays, it's a very stable molecule. So that helps. Once you do ship it to someone, not on cold chain, and it arrives, biology is incredibly good at replicating DNA, so you can just make more of it in bacteria and purify the DNA. So you effectively have an inexhaustible supply of what you've been sent. And then you can also, using the enzymes here, get DNA from other organisms and add it to the collection. If you wanted to synthesize from scratch, like what I mentioned before, where you type the bases in and it appears, that is performed by a small number of companies right now. We're probably, I'm sure that technology will get more democratized, but we're probably talking on the order of like 1 to 2 decades, I would say, before that is really distributed. But the price is coming down. They try and compare DNA sequencing to Moore's law and other kind of analogies, and sequencing definitely has reduced massively. Like for me to sequence up to say 1,000 base pairs of DNA is £3. So that's very cheap now. It used to be an entire PhD to get one sequence 20 years ago, so we're doing well. Maybe a little longer than 20 years ago. But the synthesis is sort of, it's not coming down quite that much. The other challenge is if you want to extract, so I mentioned earlier that you need little pieces of DNA that kind of select the bit that you're interested in. So they also need to be synthesized. And again, currently that is not so distributed. But again, it's reasonably quick turnaround, sort of, and like the logistics is less of a problem. It's the second question we get asked is like, can we make enzymes? Can you make primers, which are these little pieces? And we've looked into the economics, and right now it doesn't make a lot of sense because those machines are quite expensive and difficult to maintain. And the reason that the companies currently do well is that they just do a lot, like it's a volume industry. So again, it goes back to that question of like that balance between distribution and globalization, like where does it make sense? There's actually a recipe book that someone sent me, having seen a talk by me, it's like a recipe book that tries to weigh up whether you should buy or make something in the kitchen. And like for some things, it's just not worth making it, right? You just go and buy it. So we try and think of the same.
Yeah, until you have COVID, and then suddenly like the producer don't export to you anymore because they need the production for themselves.
Exactly, yeah. And this is the challenge. So I think eventually we'll see much more of that stuff. And, you know, I'm hoping other people will join and try and make those other technologies work. Because, yeah, you're right. I think during COVID the primers were less of an issue than some of the other reagents. And there were other challenges with how integrated some of those diagnostic systems are, which is, again, very non-open. That, you know, you buy your machine from someone and then you have to buy the cartridges from the same supplier, right, rather than having a kind of open platform with generic reagents, which would be better. But anyway. Oh, got one there and one here.
Can I go first? Can I? Thank you very much.
That's all right.
Fascinating talk, very new material for most of us, and you're so enthusiastic. It really looks like you enjoy your job. I have a question concerning the ethics behind all of this business. You said at the beginning, which I understand, that some people stay away from open source because they may need to or they wish to defend and control. Mm-hmm. This is true in the biotech space. Of course, we have noticed. The question is it possible that people don't engage in this and defend this activity despite the opportunities it gives to people outside the big companies and outside the dark blue piece of the world that has the money, because they see this as a dystopia. Even the fact that you start with such enthusiasm, say you can write the DNA like you write a program. Although it solves the problem of anonymization of one's individual personal DNA that would really not be appreciated by the person, this problem goes away, which is nice. But on the other hand, it looks a bit like hubris, no? It's like playing God, kind of, no?
Sure. So when I'm writing DNA, it's mostly for slightly different enzymes from a bacteria. So it's not so. So most of what we do is kind of microbial based, and then it's used for health technologies in different ways, usually diagnostic development. But you're right. There is a whole field of kind of more human based DNA interventions that brings up a lot of ethical challenges. And a lot of that. So there's ethical challenges certainly around human genome manipulation editing, and that's been around. There's a number of reports from the Nuffield Council on Bioethics, for example, about the challenges of working with human DNA and understanding that those changes that we make. So a lot of the stuff that's reached the clinic is not changing heritable DNA. So the real ethical concerns and what we don't do right now, apart from the Chinese scientists who you may have seen in the news a bit recently, is the scientific community do not edit germline DNA that is inherited by the children of the person that you've edited. What we have got in the clinic is, for example, gene therapies for eye diseases that just edit the cells in the eye and avoid the problem there, but that stays with the person that it's kind of brought on. So I'd say these are really tough issues, and there's a lot of discussion around that. What I generally try and do is sort of separate out some of these ethical challenges. So I think the whole thing around human privacy and genome privacy is one thing. The whole idea around human genome editing is a whole other set of ethics. And then the idea around kind of equitable access to health technologies is its own set. And so we try and discuss them differently, because I think if you try and intersect them too much, it gets a bit muddy. And I don't know about other people's experiences who may have been, Javier maybe has been around the open science world for quite a long time, but there was a point at which open data, which is now quite well established, was really not very well established, and there were a lot of discussions. But you often found if you went to events on open data in science, it was very easy that the entire event was around the sharing of human genomic data, which is a tiny percentage of like all of science data. So I think, I didn't go into those ethical issues, mostly because my work doesn't really sort of venture into those areas, which is not to ignore it, but I think, there are particularly there is a whole project called GP Write, which is basically writing the human genome from scratch. And that has, I think, it is a very, hubris is one word that you could potentially use. And it has a lot of attention and controversy within the scientific community as well. So I would say that, I see in this field that the process of scientific self-regulation and community kind of regulation is happening. And I think in a reasonably healthy way. I think that the other ethical concerns around access to technology maybe is a little less addressed. Like a lot of the stuff that we're doing really cutting edge now, creating therapies that cost $2 million per person, for example, is fantastic for those of us in the blue areas, but is not necessarily going to translate. So each of these different areas it's just very complex, but very interesting. So I'm happy. I think you can't join us for lunch, but it would be very interesting to talk a bit more. But so did you have a second question? Sorry, I feel like no, it was just that one. Okay, perfect.
Yeah, thank you for the talk. That was really amazing. I have just one question regarding the relationship between the different open source initiatives and the revenue. And I was wondering, is there different models that are being tested at the moment?
In the examples that I gave, or in my work?
In your world, yeah.
Okay. So in my world, which is basically the world of creating these toolkits and sending them out, so I showed that, now I've got to remember what order my slides are in. Let's see. No, not that order. Further back, there we go. Okay, so in terms of the technology side, most of that has been funded through research grants and philanthropic sources, right? So we produce the technology, we make it available to the world. Our university seems okay with that. I can't speak for other universities, right? So I'm in a quite lucky position in Cambridge in that the university itself is quite familiar and open to open technologies, and also my funder mandated it, which is always helpful. Top tip, if you want to do open source stuff, write it in the grant agreement, write it in the grant proposal, and get the funder to put it in the legal contracting. So that bit we've mostly done as a sort of like a no revenue generating exercise, if you like. And then the communities, so again, there's no real revenue there. That's more like a classic kind of network of academics. And so what we see is kind of the technology going into the community, and then people are making stuff in their own labs for their own purposes. It's sort of a very kind of decentralized grassroots model. I didn't go into the enterprise piece, but then the other question is, so there's nothing to stop you selling these technologies. That's baked in for open source. So what would it look like if we actually had production companies in these different locations that were using the open source technologies and protocols to make things and sell them? And I think that is a viable business model, partly because scientists want to do science. They don't necessarily just want to spend their time making the things they need to do the science. That's why we buy it right now. I mean, I could make the stuff in my lab. I buy it for convenience. And so we set up a trial technology transfer in Cameroon, which was successful in terms of the technology transfer, and they are selling product. So they're kind of revenue positive, but I think it's probably going to be 2 years, maybe 3, before they're really kind of sustainable without any grant funding. But Cameroon is a very specific context. so Central Africa in general is a difficult region to do business in, and it's also probably the region with the least size of market for this sort of stuff. So what we're trying to, so that was a model of basically like manufacture first. We had some funding, we wanted to test, can we transfer this technology to a challenging environment? Because if we can do it in Cameroon, we can hopefully do it anywhere. And we did, and they are moving forward. They've got a production facility. They have an innovation hub with students coming in to use the lab, and they have a number of diversified revenue streams that they're working on. We're currently, so I have a non-profit, I should say. When I say we, that started as an academic project, but it's now supported by a non-profit that I set up. And so in Kenya, we have some US aid funding, and there we're using what you might call a kind of entrepreneurship first model kind of thing. So we're basically setting up a lab, a production lab that will initially be staffed by the academic members of the initiative to supply the initiative itself, which enables us to kind of get things moving and going on. And then as part of this three-year program, there's a bio-entrepreneurship training that will happen, and also we're running biomanufacturing training. And so the people that go through the biomanufacturing training and the entrepreneurship training, we're going to identify the best ones, hopefully, and persuade them to take over the reagent production as a local business, and then test the model. Kenya is a much more conducive environment for that because it's a bigger market. It's much easier to do business there just in terms of logistics, and there's much better availability of capital. It's not brilliant, but it's okay compared to Central Africa. So that's one model there. And then a third, so I'd say we're in experiment mode, right? So we're trying to find like funding to try a few little experiments. In Latin America, it would be more of like a sales first model. So have somebody who sets up a distribution node and attempts to build a market for the reagents, and then substitutes imports with locally manufactured goods once we've figured out that it makes sense to invest in the manufacturing. So once you're making enough sales, then it makes sense to manufacture because you increase your profit margins and reduce your costs by local manufacturing. So those are the sort of three methods that we've tried so far. The one was not really a serious attempt to do a kind of sustainable business. It was more of an experiment, and it was grant funded. The second one is a bit of a hybrid, and then the third one would be more of a business proposition. But I mean, in terms of market research, everybody is annoyed with the current situation, which is great. So in terms of like a value proposition to the scientists, it's all over that. My challenge with this is that I'm not a business person. And I don't necessarily want to run a multinational import-export manufacturing business. And so we're currently trying to restructure the non-profit such that someone else does that. And I'll come back to you in 5 years and let you know if it's possible or not. But I think the advantage we have for biotechnology that is similar to the advantage for hardware compared to digital is that people do not expect like tubes of reagent to appear on their bench that they haven't paid for, whereas they do expect to download digital products that they haven't paid for. So people will pay for this, and if you can sell it for cheaper than the competitors or higher quality or faster, and the market can pay for it, then there is a business, and you can do that for less than you're actually paying, then there's a business there. And I think it really, at this point, needs someone who knows about business to make that part happen. But yeah, so I'd say we're sort of fairly confident in, and we're just at the level of scaling that right now. So we have a model for how we've dealt with the collections. We're currently going through an update to the collections. Community is getting there. We're trying to set up regional hubs for like academic distribution of the kits, and kind of put together more of a sort of regional consortium model. And then the enterprise is where we're really at the very early stages. But my take on it is that the bigger companies, I don't fully understand why they can't ship to Africa quicker than they can, because if I ship via DHL, it gets from my department to Yaoundé in Cameroon in 48 hours if I ship on a Monday. If I ship any other day, it maybe is an extra day. But yeah, it's not, like 4 weeks is a lot of time. So, I mean, to be honest, even if success for us could look like if we're successful and put these manufacturing hubs together and they force other companies to do better because there is actually a competition, that's a success for us. Whether we'll get there, I don't know, but we'll try. No worries.
Okay, thanks. I don't see any other raised hands from the Zoom participants who have not gone for lunch yet, so I guess we can wrap up.
Yeah, we went long on the questions.
Thank you. No, it's been great. Also the Q&A. Yeah. So thank you very much again for your time.
You're very welcome.
Yes.
You're welcome.